Very good morning, dear students. Myself, Dr. Arvind Singh Panwar. I am additional professor in the Department of Pharmacology, Smimir Surat. Dear friends, today we are going to study about the pharmacodynamics. As the name itself suggested, pharmaco, it means or it relates with the drug, and dynamics, it means action of the drug. So let us see फार्मेकोडायनामिक्स, इसका मतलब ये हुआ ड्रग और उसकी एक्शन या उसकी एक्टिविटी. सो लेट अस डिस्कस अबाउट द डेफिनेशन ऑफ फार्मेकोडायनामिक. सो अकॉर्डिंग टू द स्टैंडर्ड बुक ऑफ फार्माकोलॉजी, pharmacodynamics is the branch of pharmacology which deals with the study of physiological and biochemical effect of drug and their mechanism of action at macromolecular, subcellular and organ system level. So this is an overall definition of pharmacodynamics, but in a very simple way, pharmacodynamics is what the drug does to the body. मतलब एक दवाई और ड्रग बॉडी के ऊपर क्या इफेक्ट प्रोड्यूस करती है, that is called pharmacodynamics. Now, how modes of, or you can say types of drug action. So there are various modes of drug action. How does this drug act? So number one is stimulation. It is an increase in the rate of functional activity of the cell or tissue. Stimulation का एग्जाम्पल अगर हमें देखना है तो हम कह सकते हैं जैसे एड्रेनलिन अपने हार्ट रेट को बढ़ा देता है, that is called stimulation. So drug act by stimulating certain function. For example, adrenaline increases or enhances heart rate. The second one or second mode of action is depression. जैसे कि हम देखते हैं कि एसिटाइल कोलिन जो रहता है वो हार्ट रेट को कम कर देता है. इट मीन्स इट decreases or depresses the ongoing activity of the body. So that is called depression. Number three, the third mode of action is replacement. So as we see in diabetes mellitus, there occurs deficiency of insulin. So we administered insulin externally. तो इंसुलिन जो है, एक्सटर्नल इंसुलिन जो इंटरनल इंसुलिन रहता है जो नहीं बन पाता है उसको रिप्लेस करता है. सो दिस इज द थर्ड मोड ऑफ एक्शन विच इज कॉल्ड रिप्लेसमेंट. देन कम टू इरिटेशन. सो आपने देखा होगा कि अगर हेडेक होता है तो हम सिर के ऊपर बाम लगाते हैं, राइट? व्हाट विल हैपन? That balm causes irritation, mild irritation, or you can say counter irritation, and because of that the hidden part of the pain subsides. So that is the irritation or counter irritant effect of the drug. So this is the fourth mode of drug action. Then the fifth mode of drug action is cytotoxic effect. That is selective toxicity for invading parasites or cancer cells. इसके अंदर हम दो भाग में इसको हम डिवाइड कर सकते हैं. नंबर वन, एंटी माइक्रोबियल एक्शन. सो that is called AMA. It means the drug act by inhibiting or killing the microbes. That is called antimicrobial agent which act by antimicrobial action. And this effect is also produced in certain cancers, certain anti-cancerous drug which inhibit or kill the cancerous cell. So that is called anti-cancerous action, or you can also name it as chemotherapeutic effect, and the drug is called chemotherapeutic agents, right? So this is the fifth, and the last one is by modification of immune system. So you can say, जैसे हम देखते हैं कि कई बार हम स्टेरॉइड्स देते हैं जो इम्यूनिटी को कम करके ओवरऑल, जैसे ऑर्गन ट्रांसप्लांटेशन में हम लोग देते हैं तो उस तरह से ड्रग एक्ट करती है। और इन सर्टन केसेस जैसे वैक्सीन्स हम लोग देते हैं जिससे इम्यूनिटी बूस्टअप होती है और वो ड्रग एक्ट करती है, सो बाय मॉडिफिकेशन ऑफ द इम्यून स्टेटस। सो दिस आर द वेरियस मोड्स ऑफ ड्रग एक्शन। नाउ in the next topic we discuss about drug act on the body, or action or mechanism of action of the drug, right? So drug act basically, number one, outside the cell, that is also called as extracellular effect of the drug, or drug act by imparting its effect outside the cell. Then second one is the action of the drug on the membrane of the cell, and number three, action of the drug inside the cell. So let us discuss about how drug act outside the cell. So its effect outside the membrane of the cell, you can say. Number one, physical action. A physical property of the drug is responsible for its action. फॉर एग्जाम्पल, फिजिकल मास ऑफ द ड्रग, जैसे हम देखते हैं बल्क लग्जेटिव, स्पेसिफिकली इन कॉन्स्टिपेशन वी प्रिस्क्राइब दिस मेडिसिन, राइट? देन कलर ऑफ द ड्रग, सो प्लेजेंट कलर गिव प्लेसिबो इफेक्ट टू द पेशेंट। रंग बिरंगी दवाइयाँ देती है तो उसका इफेक्ट पेशेंट को जो है वो काफ़ी सूथिंग रहता है, सो डेट इज प्लेसिबो इफेक्ट। देन ऑस्मोटिक एक्टिविटी, फॉर एग्जाम्पल मैग्नीशियम सल्फेट हम एज ए परगेटिव देते हैं, वो उसकी ऑस्मोटिक प्रॉपर्टी की वजह से एक्शन होती है। सिमिलरली मैनिटोल जो रहता है वो एज एन डायूरेटिक हम यूज करते हैं बिकॉज ऑफ इट्स ऑस्मोटिक एक्टिविटी। देन रेडियो एक्टिविटी, तो रेडियो एक्टिविटी प्रॉपर्टी जो रहती है उसकी फिजिकल प्रॉपर्टी, सो आयोडीन 131 एंटी थायराइड ड्रग के रूप में यूज किया जाता है जो केमिकल थायराइडोक्टोमी करवाता है, राइट? सो दिस इज द रेडियो एक्टिव प्रॉपर्टी ऑफ द ड्रग, दैट इज द मैकेनिज्म ऑफ एक्शन ऑफ दैट पर्टिकुलर ड्रग। देन रेडियो ओपेसिटी, फॉर एग्जाम्पल बेरियम सल्फेट जो है हम बेरियम मील के रूप में यूज करते हैं, कॉन्ट्रास्ट मीडिया के रूप में डायग्नोस्टिक पर्पस के लिए हम इसको यूज करते हैं। देन स्मेल, सो वोलेटाइल ऑयल, फॉर एग्जांपल पेपरमेंट ऑयल जो रहता है ये सूथिंग इफेक्ट जो है कूल में प्रोड्यूस करता है, कूल कफ कोल्ड में प्रोड्यूस करता है। सो दिस आर द फिजिकल प्रॉपर्टी ऑफ द ड्रग व्हिच आर रिस्पॉन्सिबल फॉर देयर एक्शंस। देन सर देयर आर डेफिनेटली सर्टेन केमिकल एक्शंस आल्सो, फॉर एग्जाम्पल एंटाएसिड, सो अगर एसिडिटी होती है तो हम वहाँ पर एल्यूमिनियम हाइड्रोक्साइड या दूसरे कोई सॉल्ट्स जो है उसका यूटिलाइजेशन करते हैं, यूज करते हैं, जैसे डाइजीन वगैरह हो गई। सो ये न्यूट्रलाइजेशन रिएक्शन करवाती है एसिड और बेस के बीच में, और सॉल्ट प्लस वाटर बन के न्यूट्रलाइजेशन रिएक्शन होने की वजह से वहाँ पर एसिडिटी कम हो जाती है। सो डेट इज एंटाएसिड यूज। देन चिलेटिंग एजेंट, पॉइजनिंग के केस में हम बाल या पेनिसिलियम जो है उसको यूज करते हैं, वो एज एन चिलेट यूज करते हैं और मेटल टॉक्सिसिटी के इफेक्ट को कम करते हैं। देन एज एन ऑक्सीडाइजिंग एजेंट लाइक बीटाडीन, जो पोविडोन आयोडीन होता है, वो एज एन एंटी माइक्रोबियल एजेंट यूज किया जाता है, एंड द मैकेनिज्म इज बेस्ड स्पेसिफिकली ऑन इट्स ऑक्सीडाइजिंग प्रॉपर्टीज, राइट? Then acidification or alkalinization of body, you can say component. Certain drugs acidify and certain alkaline urine. So drug elimination के अंदर ये property जो है drug की वो use की जाती है। Then antioxidant action. So superoxide dismutase act as an antioxidant and prevent the cell injury. देन आयोनिक एक्सचेंज, सो बाइल एसिड बाइंडिंग रेजिन्स जो रहती है वो आयोनिक एक्सचेंज के रूप में यूज की जाती है। सो दिस आर द केमिकल एक्शन ऑफ द ड्रग। सो दिस टू आर द बेसिकली फिजिकल एक्शन और द एक्शन बेस्ड ऑन द फिजिकल प्रॉपर्टी ऑफ द ड्रग। देन कम टू ड्रग that can act on the cell membrane. So cellular action of the drug, number one, they act by acting on the specific receptor. So specifically when we talk about the agonist and antagonist, they act by binding with specific receptors like adrenaline and propranolol. So adrenaline is an agonist and propranolol is the blocker for that particular receptor on which adrenaline act. So they act as an adrenoreceptor, and acetylcholine and atropine act on the cholinereceptor, like that. Then acting on the specific ion channel by inducing number one, opening of the ionic channels or closing of the ionic channel. For example, local anesthetic agent. So सोडियम चैनल्स जो रहती है वो सोडियम चैनल ब्लॉकर्स जो हैं वो हम यहाँ पर यूज करते हैं. Then inhibition of the membrane bound enzyme and pump. So cardiac glycoside ये आप लोग लेक्चर में पढ़ेंगे. डिजॉक्सिन एंड डिजिटॉक्सिन they inhibit membrane bound sodium potassium ATPase enzyme, right? So drug may act by this mechanism. Then imparting physiochemical interaction, you can say saturation in biophase. So general anesthetic agent जो है वो physiochemical interaction के modulation से या method से जो है वो act करता है. So this is the second part where drug act on the cell membrane. Then drug can act within the cell. So intracellular effect of the drug. So by interfering with various metabolic process. Number one, enzymatic induction. That is also called as stimulation. Example, एड्रेनलिन जो रहता है वो एडिनाइल साइक्लेस को स्टिमुलेट करके एक्ट करता है. So that is called enzymatic induction. Similarly, enzymatic inhibition, here there occurs inhibition of the enzymes. Example है phenol या aldehyde जो है जिसमें inhibit enzymes non-specifically. Physostigmine and neostigmine compete with acetylcholine for cholinesterase. All these things will be discussed later. We'll discuss it in the next chapter. Then inhibition of the transport process. So drug may act by inhibiting this transport process. For an example, proben acid block secretion of penicillin in renal tubules. Then incorporation into larger molecules and nucleus. For an example, specifically when we talk about the sulfur drugs, they resembles para-aminobenzoic acid PABA and therefore incorporated in the synthetic process of folic acid in place of PABA. And similarly 5-FU incorporate into mRNA in place of uracil. So this is basically the mechanism of action. Then by altering metabolic process, example है beta lactam antibiotics act by inhibiting cell wall synthesis in the bacteria. Similarly sulfonamide act by inhibiting folic acid synthesis in bacteria. Then we'll discuss about the targets for the drug action. So number one is the receptor. So receptors, they are the specific drug binding site in a cell or on its surface which mediate the action of drug. They are macromolecular proteinous particles. They are located on the membrane in the cytosol. You see in the figure, these are different type of receptors. In the first diagram, you see agonist bind with the receptor and produces various effect by transduction mechanism, which ultimately leads to either enzymatic activation or inhibition or ionic channel modulation or DNA transcription. So this is the indirect method, and sometimes it may act directly also. Whereas antagonist bind with this receptor and block the action of agonist on that particular receptor. So ultimately there will be no effect, no endogenous mediator is blocked. Second one are the ionic channels. Here you see ionic channels, ion channels, where blockers and modulators. So blockers get blocked that ionic channel and permeation get blocked because of that blockers, and modulators, when they bind with these ionic channels, either they increase or decrease the opening probability of the channel. Then come to enzymes. Now enzymes play an important role in regulating a number of body functions like synthesis of various metabolites required for cell growth. synthesis of secondary messengers like cyclic AMP and GMP, transport of ions, synthesis and hydrolysis of various neurotransmitters, etc. Therefore, enzymes are the important target for many drugs. On binding with enzymes, drug can either decrease or increase the rate of enzymatically mediated reaction. As you see, inhibitors when blocked, the normal reaction get inhibited, then false substrate, the consequence are abnormal metabolite production, then prodrug, so ultimately active drug is produced. Now, two type of inhibitions are there. One is competitive inhibition and second one is non-competitive inhibition of the enzymes. So let us discuss about the competitive inhibition, which is again subdivided into equilibrium type of competitive inhibition and non-equilibrium type of competitive inhibitions of enzyme. So, when we talk about equilibrium type of competitive inhibition, in this case, drug compete with the normal substrate or coenzyme, whereas in non-equilibrium type of competitive inhibition, drug bind with the same catalytic site of the enzyme but with strong covalent bond, and therefore substrate is not able to displace it. In the equilibrium type of competitive inhibition, the rate rate kinetic increases, Km increases with Vmax remain unchanged, whereas in non-equilibrium type of competitive inhibition, Km equilibrium constant increased with the Vmax decreases, but Vmax decreased. Third, the equilibrium type of competitive inhibition, higher concentration of the substrate is required to achieve half Vmax, whereas in case of this methotrexate is the example which compete with the DHFA for dihydrofolate reductase, and here the example is physostigmine and neostigmine in the equilibrium type which compete with acetylcholine for cholinesterases. So these are the things. But when we compare it with non-competitive type of inhibition, here the drug bind at the adjacent site, not at the catalytic site, adjacent site of the enzyme, and not at the catalytic site, and ultimately alter the enzyme in such a way that it loses its catalytic property. The equilibrium constant remain unchanged, but Vmax is reduced. Example is digoxin inhibits sodium potassium ATPase and aspirin inhibits cyclooxygenases. So these are the enzymatic inhibition, example of enzymatic inhibitions. Then come to carrier molecule. The drug may be carried by transporters. You see in the first diagram, normal transporter, the drug bind with certain sites, and then ultimately it transported from outside the cell to inside the cell. Certain inhibitors are there which ultimately inhibit or block the binding of this transporter, binding site of the transporter, and ultimately inhibit the transport. So sometimes false substrate also causes abnormal compound accumulation, right? So this is about transporter. Now come to the most important prospect of the pharmacology, or you can say the mechanism of pharmacology, and that is the receptors. So first of all, we discuss about the history of the receptor. So the side chain theory of the receptor is proposed by Paul Ehrlich in 1845 to 1950. J. N. Langley is a scientist from 1852 to 1925. In 1905, proposed a drug might act upon spatial site or receptive substrate SAR. Then in 1937, a scientist named Clark proposed receptor occupational theory. According to this, A is the drug, R is the receptor. So when both bind together, the produces K, means drug receptor complex. Two more scientists, Arians and Stephenson, in 1950 introduced the new concept regarding affinity, efficacy, which is also called intrinsic activity, and about spare receptors. J. H. Goodham and H. O. Shields also contributed in the development of dose response relationship concepts. Now, two-state receptor model, which is a very popular model and stand for very long period of time. Now, according to this model, the receptors are present in two interchangeable states, that is RA and RB, which are in dynamic equilibrium with each other. So, RA and RB are the receptor which are in dynamic equilibrium with each other, and drug bind with this, or not only drug but antagonist also bind with this and ultimately produces the effect. So here you can see 7 alpha helical structure of the receptor. Then the topmost part is the ligand binding domain, and the lowermost part is the transduction of the signal into responses. Now, here you see catecholamine, which is a non-adrenaline the first messenger beta receptor that increases the activity of cyclic AMP. When bind with the receptor, produces increase in the activity of cyclic AMP, that is called second messenger, which ultimately modulates the activity of several enzymes, and ultimately the cell response or the effect of the drug produces. So this is the cascade of the mechanism of action of the drug through receptor. For example, the drug bind with the receptor on the first point, and lastly the cell response produces. Now, the function of receptor: recognition of a specific ligand molecule, that is the very important function of receptor, whether it is agonist or antagonist. Receptor recognize the ligand. Then transduction of signals, it is propagation of the signals from outside to inside of the effector cells, and second one is the amplification of that particular signals. Then integration of various regulatory signals, for example extracellular and intracellular. Then maintenance of homeostasis by adopting process of receptor regulation, upregulation and downregulation. So these are the two important phenomenon: receptor regulation. First one is downregulation. So what is downregulation? Continuous and prolonged exposure of agonist to tissue receptor leads to decrease in the number of receptor. अगर बहुत लंबे समय तक एगोनिस्ट का एक्सपोजर कराया जाए रिसेप्टर को, तो रिसेप्टरों की संख्या कम हो जाती है या उसकी सेंसिटिविटी कम हो जाती है. इसी फिनोमिनन को हम कहते हैं डाउन रेगुलेशन. So tachyphylaxis develops because of this. Loss of efficacy with frequently repeated doses, as in asthmatics who uses salbutamol chronologically on long-term basis. This is because of downregulation. Upregulation: when tissues are continuously or prolonged exposed to an antagonist leads to formation of new receptor. अगर हम रिसेप्टर को लंबे समय तक एंटागोनिस्ट के प्रति एक्सपोज रखेंगे, तो वहाँ पर कुछ नए रिसेप्टर जो है वो रिप्रोड्यूस हो जाएंगे, और रिसेप्टर की संख्या जो है उसमें भी बढ़ावा होगा, वो भी बढ़ जाएंगे, और इसी फिनोमिनन को हम अप रेगुलेशन कहते हैं. दिस इज कॉल्ड अप रेगुलेशन. सो precipitation of angina attack on sudden withdrawal of beta blockers, and it's because of the secondary prophylactic agents. It's because of the upregulation of the receptor. So these are the two kind of drug regulation: upregulation and downregulation. Then type of receptor. So basically three types of receptors are there. Number one, physiological receptors. So basically the receptor which participates in the binding of the ligands, that is called agonist or antagonist, and because of which various function incorporated in the body are called physiological receptor. Silent receptors are the receptors which are located in the body but no agonist antagonist bind with these sites, and they are called silent receptors. Spare receptors means after binding of the drug with this specific receptor, signal transduction occurs and ultimately pharmacological effect develops. So this is about the spare receptors. Now drug action and drug effect. There is difference between drug action and drug effect. So binding of the drug to its specific receptor is called drug action. ये बहुत important point है. Drug action और drug effect में बहुत difference होता है. एक ड्रग स्पेसिफिक रिसेप्टर पे सिर्फ अगर बाइंड भी कर जाती है तो हम बोलते हैं ड्रग एक्शन. दिस बाइंडिंग ऑफ द ड्रग टू द रिसेप्टर कैन लीड टू either conformational change in the receptor, in case of agonist where action is produced, and prevention of the conformational change on adding agonist in case of antagonist. So चाहे एंटागोनिस्ट बाइंड करे रिसेप्टर को, चाहे एगोनिस्ट बाइंड करे, सिर्फ बाइंडिंग जो ड्रग की होती है रिसेप्टर पर उसको हम बोलते हैं ड्रग की एक्शन. And the ultimate effect brought about as a consequence of the drug action means because of the binding of the drug to the receptor through a series of intermediate steps is called ड्रग इफेक्ट. सो एक ट्रांसड्यूसर मैकेनिज्म के माध्यम से ओवरऑल बिकॉज ऑफ बाइंडिंग ऑफ द एगोनिस्ट और एंटागोनिस्ट टू द रिसेप्टर एक फाइनल आउटकम जो प्रोड्यूस होता है उसे हम कहते हैं ड्रग इफेक्ट. सो दिस इज द बेसिक डिफरेंस बिटवीन ड्रग एक्शन एंड ड्रग इफेक्ट. नाउ देयर आर इंपॉर्टेंट टर्मिनोलॉजीज. नंबर 1, affinity. So it is the ability of drug to combine with the receptor. So affinity को हम ऐसे भी बोल सकते हैं जिस प्रकार से एक गम की बाइंडिंग कैपेबिलिटी रहती है, उसी तरह से एफिनिटी जो है एक ड्रग की रिसेप्टर के साथ बाइंडिंग कैपेबिलिटी जो रहती है, उसकी जो एबिलिटी रहती है, उसको हम एफिनिटी बोलते हैं. Intrinsic activity is also called as efficacy. So it is the ability of drug to bind with the receptor and to induces the response. So ये उसके आगे क्या प्रोस्पेक्ट है. एक बार ड्रग रिसेप्टर से बाइंड करके कितना इफेक्ट प्रोड्यूस कर सकती है, ये जो एबिलिटी ऑफ प्रोड्यूसिंग इफेक्ट आफ्टर बाइंडिंग विद द रिसेप्टर जो है उसको हम बोलते हैं इंट्रिंसिक एक्टिविटी. Then come to agonist. So agonists are the substances which bind with the receptor and produce a certain effect. So not only certain but maximum effect, or you can say they have both affinity and maximum intrinsic activity. 1 equal to 1 मतलब ये एगोनिस्ट जो सब्सटेंस रहते हैं, नंबर 1 ये ड्रग जो है, ये एगोनिस्ट जो है ये रिसेप्टर के साथ बाइंड होंगे और बाइंड होने के साथ साथ इंट्रिंसिक एक्टिविटी भी मैक्सिमली प्रोड्यूस करवाएंगे. ऐसे सब्सटेंस को हम एगोनिस्ट बोलते हैं. Partial agonists are the substances which bind with the receptor and produces some maximal effect but antagonize the action of the full agonist. मतलब partial agonist वो substance होते हैं जो receptor के साथ bind तो agonist जैसे ही होते हैं पूर्ण रूप से, लेकिन जो intrinsic activity रहती है वो agonist से थोड़ी कम produce करवाते हैं. सब maximal effect जो है वो produce कराएंगे और दूसरी तरफ ये agonist को receptor से bind करने से रोकेंगे भी. So this is called, they are called partial agonist. Then come to inverse agonist. So these are the substances which activate the receptor and produces effect in the opposite direction of that of agonist. मतलब ये inverse agonist जो रहते हैं ये receptor पे bind तो होंगे लेकिन जो effect produce होगा वो agonist के बिल्कुल opposite, बिल्कुल विरुद्ध direction होता है, opposite direction जो होता है उसमें इसका action produce होगा. So they are also symbolic represent as minus I max. Then antagonist. So antagonist are the substances which bind with the receptor. ये receptor के साथ bind तो होते ही हैं. They have full affinity. They occupied them. ये वहाँ पर occupy कर लेते हैं receptor को, but does not produces any response. मतलब बाइंड तो करते हैं लेकिन रिस्पॉन्स जो रहता है वो जीरो रहता है. इंट्रिंसिक एक्टिविटी जो रहती है वो जीरो रहती है. Therefore they also prevent the action of agonist तो ये बेसिकली रिसेप्टर को ब्लॉक कर देते हैं, एगोनिस्ट को भी रिसेप्टर से बाइंड करने से प्रिवेंट करते हैं. So that's why they are called antagonist. And agonist, antagonist, they are the substances which act as an agonist at one side while act as an antagonist on another side. So this is about an agonist, antagonist. Now come to dose response relationship. So whenever we administer or give any dose of the drug, it definitely produces certain response. So it has two component, dose, that is plasma concentration relationship, and second one is plasma concentration relationship and response. When a drug is administered systemically, generally the intensity of the response increases with the gradual increase in the dose. So the response taken on the dose taken on the log x-axis and the response measured in millimeter on the y-axis, and the graph between these two is called dose response curve. So it is the curve obtained by plotting log doses on x-axis and its consequential responses plotting on the y-axis. The curve becomes sigmoid and a linear relationship between the log of the doses and the response are seen in the intermediate 30 to 70% response zone. So dose response curves are useful in number one, bioassay, estimation of the biological component. Number two, to calculate the therapeutic index of the drug, that is LD50 by ED50. Then to compare the potency and efficacy of the drug. Then to study drug antagonism and competitive antagonism, the curve shift to the right and run parallel. In non-competitive antagonism, the curve become flattened and efficacy decreased. Thank you. So this is the first lecture. Tomorrow, day after tomorrow, we'll discuss about the second lecture, continuation of the pharmacodynamics. Thank you very much.